Mechanism of Muscle Contraction. Mechanism for the contraction and relaxation of a sarcomere is called the sliding filament theory. During a contraction, sarcomeres become short. However, the actin and myosin filaments remain the same size. They just slide past each other, changing their relative positions as the muscle contracts and relaxes. An action potential must be triggered to contract from somatic motor neuron, which reaches to neuromuscular junction. It is a junction between the neuron and the skeletal muscle. Due to the depolarization of axon terminal, it releases neurotransmitter acetylcholine into the synaptic cleft between the neuron and muscle. The muscle fiber fire an action potential of its own as neurotransmitters trigger it. This electrical signal passes through muscle fibers, plasma membrane, and it also passes into the plasma membrane's tubular invaginations called T-tubules. Sarcoplasmic reticulum is an organelle that consists of a membranous network. Sarcoplasmic reticulum stores calcium ions and then isolates them from the rest of the cytoplasm. As the action potential passes through the T-tubules, the sarcoplasmic reticulum releases calcium into cytosol. The calcium ions diffuse into the cytoplasm of the sarcomeres. Within the sarcomeres, the actin filaments are two strings of pearls twisted around each other. The myosin filaments are thicker than the actin and have knob-like heads at their ends. Troponin and tropomyosin lie on the actin filaments, whereas myosin heads bound to the molecules of ADP and inorganic phosphate. As the sarcoplasmic reticulum has released calcium, the calcium ions bind to the molecules of troponin. After binding, troponin shifts its own position and pulls the tropomyosin molecule aside. When tropomyosin moves, myosin binding site on each of the actin subunits of the actin filament gets exposed. When a myosin head binds to actin and forms cross bridge. After binding, the molecules of P, I, and ADP are released. The release triggers the myosin heads to pivot, forcing the actin and myosin filaments to slide past each other. This movement represents the power stroke of muscle contraction. For additional power strokes, myosin must first bind to ATP. When ATP binds to the myosin heads, the heads release the actin filament. ATP is quickly hydrolyzed into ADP and PI. The energy released by hydrolysis prepares myosin head for another cycle. As the action potential decreases, calcium levels drop, and tropomyosin again hides the myosin binding sites. Actin and myosin filaments slide back to their original positions. The sarcomeres of a muscle fiber contract and relax with each signal to form a motor neuron.
For more such videos on all subjects, practice questions and a lot more, download iPrep and learn unlimited.